Well, hello everyone. Sorry I couldn't be there today. My daughter is sick, and hopefully you are behaving stupendously for the lovely guest teacher over there. Previously on Biology Honors, we watched a video and talked about how we can solve the crime. Then you looked up PCR, and hopefully you got yours stamped at the door. And now hopefully you just got done doing this. And without seeing your answers, I'm going to try and guess that for this first one, you talked about how, let's see, because it's tiny, that's why we have to do it in a tube, because DNA is so small. And what process is he mimicking? That's going to be DNA replication. Now that little quarter sheet of paper, I want you to glue it onto your thinking page somewhere. I don't know if you'll put the whole page on a left, on a right, half on a left, half on a right. That's something that you need to decide. So let's talk a little bit about DNA replication. It's all about making more and more and more and more copies. And Mullis got the Nobel Prize because he figured out how to do that in a test tube. Whereas what we're going to learn about is what your cells do every day. All right, so DNA. One of the cool things about it is it can replicate itself. Not anything else can do that. The ribosomes don't, the Golgi apparatus doesn't, the plasma membrane doesn't. DNA is the only thing. And the reason why it can replicate itself is because of its structure. And hopefully you've got this down. We know we love DNA because it's made of sugar, phosphate, and bonded down one side. And we know adenine make a lovely pair. Cytosine would feel really bare. Now, this, hopefully you saw from making your model that when you made your model on the one side, when you went to do the other side, the only way to make it work was to have them go backwards. In science, that buzzword is anti-parallel, something you might want to add to your notes. The other thing we need to talk about on DNA, which we haven't before, was location. Location, location, location. Where are things bonded on the molecule? Now, we've got this guy right on over here to show you, and it can get a little confusing, but well, it gets confusing because you haven't learned about how to number, how to draw these things. That's stuff you're going to do next year in chemistry. But what this structure represents is an oxygen bonded with a bunch of carbons. And as you get up in higher levels of science, we get lazier and lazier, and we don't want to have to draw all these carbons. So instead, we say, forget that. I'm just going to number them. Since they're all carbons anyway, who cares? And in numbering, they start at noon, which in this case is the oxygen, so they're not going to number it because they only number carbons. And then they go clockwise, so then you've got 1, 2, 3, 4, and 5 is hanging out there. Now for us in bio, those are the only ones that we care about. You want to remember that 5 is on the phosphate. We want to remember 5 is over here. It's what's attached to the phosphate. And 3 is down here. We're going to care about that later. But because of it being like that, and this DNA going in this direction, the top one over here, we call that the 5' side of the DNA. And down here, since this guy right here is the bottom, we call the 3'. And again, because these guys are backwards, if it's 5' on this end, we're going to call that the 3' end on that side. If it's 3' on this end, we call it 5' on that side. That's all you need to memorize is for the DNA when it talks about location. The 5' end and the 3' end. And you can take that DNA, and if you write 5' over here, we know it's 3' over there. And if this guy's the 5', that means down here is the 3'. And if this guy's the 3', that means down there is the 5'. So look at this pattern. Maybe draw it on the side of your notes. Not your left-hand side. We're going to do something with that later. But this is one of the big things, the next level of DNA structure. You got sugar phosphate in a base. Time to go to level 2. All right. So DNA replication, whether you're a prokaryote or a eukaryote, it's pretty much the same. Hopefully, you remember those words. If not, a great study technique, go back in your notebook and look them up. All right. Now, DNA replication is considered a semi-conservative process. There's another vocab word to highlight and memorize. We're not going to go over the different, what we thought it was, type processes, but that's more AP Bio. But in this class, we're going to know the one that actually worked. And what that means is you've got your old strand, aka the parent strand. It's going to get broken in half, right down between those bases, and the 1 is going to become 2, and that daughter strand, or the new one, half of it is from the original, and the other half of it is brand new. That's what it means for semi-conservative. All right, so let's talk about replication. The first step, everybody's favorite step, step 1. When helicase is going to go and unzip that DNA, unzip your genes. Everybody always remembers helicase, as much as you remember that the mitochondria is the powerhouse of the cell. So helicase is going to come and rip it apart. But the problem is it wants to come back together. So that's going to lead us to number 2. Hold on, pardon me just a moment. I have to take my dog out to go potty. Okay, sorry about that. So step 1, helicase rips the DNA apart. But it will come back together unless we prop it open. So that's where these guys come in. The SSBPs. Think of them as a door jamb, something to wedge in so that the DNA can't come back together. Now, you can memorize single-stranded binding proteins, but on the quiz, on the test, SSBPs are totally fine to use. But that's what your door jamb is. And it's going to keep that DNA separated. Now that the DNA is wide open, we got to get to copying. But we can't copy just yet because DNA polymerase, well, he's not that bright. He needs to know where to go. And that's where this primase is going to come in. And primase is going to put down a little primer. It's going to attach it to one of those nucleotides. And that primer that it attaches is kind of like the lights at the airplane, at the airport at night. It's going to tell DNA polymerase where to land. So helicase unwinds, SSBPs holds them open, primase puts a primer down. Now we can finally do something. Now that it's propped open and DNA polymerase knows where to go, it's going to attach. And oh, I just said that. So there's what I said in words. Helicase unwinds the DNA. SSBPs keep it open. Primase puts down a primer. Now DNA polymerase can do its job. And you don't have to write out the word DNA polymerase. You can totally abbreviate it DNA poly. And what DNA poly is going to do is it's going to add the complementary nucleotides. So if it sees an A, it's going to attach a T, and so forth and so on. But there is a problem. So you can see here, the DNA is opened up, helicase is opening up, primase is going down. This DNA polymerase right here on top, well, it's following right behind helicase, no problem, uninterrupted. It just keeps going until helicase stops, then it stops. No problem whatsoever. But the problem is DNA polymerase can only add, how I remember it is A, D, D, 1, 2, 3. It can only add to the 3' end. So DNA polymerase attaches here, and now it can add that way. And it just keeps going, no problem. Well, on this bottom strand, there's not a 3' there. The 3' is on the other side. So once it opens up, well, there you go. There's our 3' end. DNA polymerase is going to go anti-parallel because that, oh man, it just opened up some more. And there's new primer. Son of a, I got to go back. And it's going to do there. And it does, oh wait, crap. It opened up some more and laid down some new primer. Now I got to go back. So because it has to keep going back, it's going to do stuff in fragments. And the fragments are named after the gentleman who discovered them. They're called Okazaki fragments. I think the spelling on it is like the next slide, so you don't have to worry about writing that yet. But it takes a little while. It's going to fall behind. If this were a race, it's going to slow down because it has to keep going back. So how we distinguish, how we label the differences is the one that doesn't have to stop, we call the leading strand. And the one that has to keep going back and fixing things, because helicase keeps opening up more, is the lagging strand. And what I've found is, for DNA replication, this is usually the hardest thing to understand. And tonight's homework, you'll be watching a video, and if you're not getting it from me, hopefully they will explain it in a different way with better animations so you understand it, because that's your goal. This is the hard part. What's the difference between the leading and lagging strands, and what does Okazaki fragments have to do with it? All right, so DNA polymerase adds nucleotides continuously. That means non-stop. It just keeps going to the leading strand because it's following helicase. Now, for the lagging strand, it's got to go back, and now that there's more open, it's got to go back So it's going to add it in fragments. And those fragments, oh, it's the next slide, I think. Okay. So again, okay, DNA polymerase is adding. Hopefully you got that. If you don't have that by now, we got bigger problems. It's going to add and make those Okazaki fragments. There we go. Okazaki fragments. Those Okazaki fragments are only on the lagging strand. You have a continuous fragment. Well, no, you have a continuous strand on the leading strand, and the lagging has the Okazaki fragments. But now that there's pieces, we got to stick those pieces back together, and that's ligase. Ligase is the glue that puts those Okazaki fragments together. So here's the video. There's helicase right there. Keeps opening it up. Here comes DNA polymerase on the leading strand. It's just going to keep following right behind helicase. DNA polymerase on the lagging strand is going to do its job. It's going to attach, and by the time it gets done, and don't worry about the numbers, that's AP Bio. By the time it gets done, oh crap, here's a whole new section that's opened. I got to do it again. I got to go back and do it again. All right, so I'm just going to let you sit and stare at this for a moment. I'm going to try not to do the football marks on it. Again, don't worry about DNA polymerase, the Roman numerals after it. That's AP Bio. All right, so watch the picture. Helicase is moving on down. DNA polymerase on the leading strand is following right behind it. DNA polymerase on the lagging strand is going its proper direction. And it's all good until helicase has moved down some more and opened up a section. So now primase has got to reprime it so DNA polymerase can attach and make a new Okazaki fragment. All right. Now we need to make sure everything was done correctly, because if DNA polymerase messed up and attached a C to an A, we have a problem. Think you call that, oh yes, a mutation, which is next unit, I think. It's coming up, and that can be a problem. So DNA polymerase is going to go back and double check that it did its work correctly, like any good student. And once everything's good, it's done. And now when you started with one DNA, you now have two that are identical to what you started with. It's like you took them to the copy machine and hit copy. You started with one, and now you have two. And they look exactly like the ones you had before. So one goes in, two come out. So I want you to take 5-ish minutes. You can work with your table buddy, your shoulder partner, your entire pod, and see if you can write down, without looking at your notes, what each one of these did. We just talked about them. It's still fresh in your brain. If you can't remember, see if your buddy does. Do you remember? And if your buddy doesn't, then ask your other table buddy. And if they don't remember, then you got one more to choose from, hopefully. And if they don't remember, then pull out your notes. So again, take about 5 minutes. When you're done with that, come on up and get the next piece of homework. It's a packet that looks like this, only yours is black and white. After you've got the players, what you're going to do is you're going to model DNA replication. This is going to go on your next right page. Hold on just a minute. Here we go. Slideshow. All right. Oh, hold on. Let me switch these all around. There we go. So, hopefully you've done this. Oops. Chihuahua. Let me get my poop together. You've done this. You did your notes. We just did that. Again, this side, leave blank for now. We'll deal with that later. You're going to pick up hold on, I'm multitasking, not very well. There we go. You're going to pick this up. You're going to trim around the black thick line. The dotted line is for folding. Then you're going to take your scissors and you're going to be DNA helicase and you're going to unzip those jeans. Then you're going to fold it open. And that's just going to, and by holding the creases down so it'll stay open, now you're being an SSBP. Then you're going to trim this out, glue that in on the inside of the doors, as you can see over here. Color it, make it nice. Add the new strands, the leading and the lagging. The labels are on there. You're not going to be able to make your Okazaki fragments because it's too big, but you can start getting an idea. Then once you finish that, answer the questions on the worksheet. Once you finish that, come on up and get a stamp. And we will go over it on Tuesday. Once you finish that, you can pick up the homework, which is the Amoeba's Sisters video. Now, you're going to watch the video. Most of you are probably going to take notes at the same time. That's fine. That's going to go on the next right page. What I want you to do on the left-hand side is, okay, this was kind of like a summary of the video. Now I want you to do a cliff notes of the summary. Recap the recap. You can do bullet points, you can do a mind map, you can do doodle notes. But you've heard me talk about it, you're going to watch a video on it, and then you're going to write about it. And hopefully by Tuesday, you've got this DNA replication stuff down that if I were to give a pop quiz, maybe, you would be totally ready for it. All right, so we're going to, my dear guest teacher, if you would be so kind as to pause the video right now, that will be up there for them so they know what to do for the homework. If they want to take a picture of it, it's totally okay. My notebook is up front as an example if they want to see it. They can take a picture of it if they want. That's totally okay. When you are done with this worksheet up here and you come up to get your stamp, you can check your answers. You cannot take a picture of the key, but you can check yourself that you did it correctly. All right, so, oops, wrong PowerPoint. So again, these guys, take 5 minutes, do your summary. Then pick up your homework, the classwork, pick up the model. Then get that stamped and pick up the homework. So, guest teacher, if you would be so kind as to pause it now. See you guys later. Bye.